Now here you see in today's topic we'll be dealing with genetical disorder. Now what are genetical disorder? In the earlier topic we had done the mutation, but any uh, problem or any missing or anything in the chemistry of DNA may lead to a genetical disorder. So for studying the how do we detect that this is a genetic disorder? Now you see the study of the family history and the inheritance of the particular generation of a family is called pedigree analysis. So what is pedigree analysis? You have to study the family history. Okay, family history, and I'll show it to you how do we study that family history and why is it important. It is a strong tool. Tool means it is very essential to study. See, to trace the inheritance of a specific trait. For example, if a child develops certain abnormalities, if nothing is found, so that means it has to be some genetical disorder. Then it has a specific trait. Secondly, a disease can be found, and there are certain diseases that is linked with X-linked diseases and Y-linked diseases that we will be dealing in the later part. X-linked means these diseases happen only to the female. This diseases happen only to the male. That means what? XX. That means it is in the homozygous condition and XY in the heterozygous condition. So if X is the carrier, so it may represent in the homozygous condition or it may not represent in the homozygous condition. It would be a carrier and it would be represented in XY condition. Okay. So we will be dealing it. Just an example. It's not in your syllabus. So don't worry if you don't understand it. Now here, if you see, focus here. Now here is just an example of pedigree analysis. You see, for example, the square, there are certain color codes and symbols taken together. The square one is supposed to be the male, the round one is a female, and the diamond one, the diamond shape one is basically this is an unexpected, unspecific. So you have 3 dots like this, that is known as affected individual. Now here again you find the traits. See, these are not there, and again it is found in the later generation. So this is how a pedigree analysis is done. Now if we focus here, we see that Mendelian disorders. Mendelian disorders means what? It is an alternative or a single gene mutation. That means, for example, alternate gene may mutate, a single gene may mutate, and that can be studied through pedigree analysis. Now, there are certain diseases, that result due to Mendelian disorder. Now, the first one is the hemophilia, the cystic fibrosis, the sickle cell anemia, color blindness, phenylketonuria and thalassemia. See again the X-linked and the Y-linked disorders. Now basically you see these genetic disorder basically has no medicine to cure. Okay, so here if you can focus here, you see again it's the same example. See, the male is affected, the female is not affected. So if the male is here again it comes here, and this one, these are the affected, and another one is this is for sickle cell anemia. Okay, so these are the simple pedigree analysis. You go through it, you can very well understand. Now again you see, so this is now if we go through hemophilia, what is basically hemophilia? See, this is a sex-linked recessive disease that shows the transmission from unaffected carrier female. That means female, that is XX in the homozygous condition. If one of the X is carrying it, in homozygous condition, it will not represent the disease. But what will happen? It would be seen in the son, because the X and Y, the X is affected. So in the heterozygous female, it for hemophilia may transmit a disease to the son. The possibility of the female producing an hemophilic is extremely rare, because mother of such a female has to be at least a carrier and the father should be hemophilic. So that is the case. Now you see in sickle cell anemia, what happens? You see the hemoglobin. In the general condition, it is a heterozygous condition, and it is HBA, HBA and HBS. Now on the three possible genotype only, the homozygous individual of HBSC, this is the condition, shows the disease of the phenotype, and the homozygous individuals, this is the carrier. So A and S. So in SSC, this is the condition. Now in this genetic condition only the disease will be caused. Okay, homozygous condition. Now this defect, how can this defect? This defect can be a substitution of the glutamic acid. That means a protein coding it. You see, in normal hemoglobin, it is, these are the codes, nucleotide codes. This is also a nucleotide code. See, now since it's normal hemoglobin, it is coding for glutamic acid. But here, since the code is different, it would be coding for a different C. Instead of GLC, VAL is there. So that is how the sickle cell anemia is caused. Okay. Now see, valine in the 6th position, that means if the DNA sequencing is like that, the chain would be completely different. So you see the substitution of amino acid in the globin protein present due to a single base substitution in the 6th codon of beta globin is GCA. This is the code, that is there. And phenylketonuria is the, see, it is an, what is this? This is the enzyme that converts amino acid in the phenylalanine into tyrosine. So what has happened? As a result, the phenylalanine is accumulated and converted into phenylpyruvic acid, another DNA. So that is a metabolical disorder. This is inborn error of metabolical disorder. This happens, the protein that is required for the body is completely lost from the urine. This is a disease, and persons have to give in blood transfusion or extra protein in their diet so that the body is able to maintain its normal metabolism rate. Okay, now you see, now chromosomal disorder. Now next comes the chromosomal disorder. That means it is either an absence or presence of an extra number of chromosome. For example, it may be one is absence and presence, that is chromosomal disorder. It can be detected as aneuploidy and polyploidy. In aneuploidy, what happened? You have these are responsive autosomes and sex chromosomes, we have already said. So these are not autosomal disorder. Okay. Now here either the person gets X chromosome, 2X chromosome, that means they may be super females, that is known as Klinefelter syndrome, XXXY, and another may be X0. That means these are Turner syndrome. These are basically individuals. They will have this Turner syndrome. This will have rudimentary ovaries, no secondary sexual characters. Okay. And these female, they'll be female but without showing any female characters. And Klinefelter syndrome is, they'll be male showing the female characters. And Down syndrome is also known as Mongolian idiocy. We'll be dealing with later. And the last part is known as a polyploidy. What is polyploidy? The full set of autosome increases. Okay. So this, if the full set of autosome increases, that means they'll have a very complicated genotypic and phenotypic characters. Basically this polyploidy we find only in the plants. Okay. Now here if you focus on the screen, see, we'll be dealing with, first, you see this, it is individual of Down syndrome. This is also known as the Mongolian idiocy. Okay, now since and it's a phenomenon of polyploid, the condition is seen in the plants. Okay, now you see, instead of 43 chromosome, Down syndrome, we have a 23 pairs of chromosome. Okay, but here, the individual gets 21. That means 21, 21 and 21, 321 series. The affected individual, short stature, round neck, furrowed tongue. They can roll the tongue in a furrow and partially open mouth. The palms are very broad and they have got certain physical, psychomotor and mental development is retarded. So it is also known as a Mongolian idiocy. Klinefelter, I said, these are super females with XXXY. So they have got a 47 karyotype. Normally, individuals have 46 chromosomes. So since they have got an extra X chromosome, so they are super females, XXY. They are overall masculine because presence of Y gives a masculine character, but extra X, say double X condition, these basically happens due to non-chromosomal disjunction. That means what? During the telophase or when the chromosomes separate, they don't separate. So that is also a mechanical failure. Now in Turner syndrome, you see this due to the absence of one. So they are X0. Such females are sterile. Okay, this was also, I had dealt it earlier. Okay, Turner syndrome, Klinefelter syndrome. So you go through them. Okay, now with this we come to the end of the